All right, hello everyone. So this week we are going to start the ecology portion and we are looking at the first abiotic factors lab. So in this lab, we're going to focus on abiotic and biotic variables and how they influence the environment. As we're doing this, we're going to learn about some data collection tools and how to analyze our data. Since we are moving into the ecology portion, I want to introduce to you what is the study of ecology. So this is when you're looking at the interactions between organisms and their environment. So although that seems pretty straightforward, it's actually a little complicated, much complicated to be precise, because how can you really measure the influence between organisms and their environment? But essentially, that is what ecology is. So if we take a step back, we can take a look at the environment and break it up into two variables. We have the biotic factors. This is going to be the living components in the environment, and the abiotic factors. These are the non-living variables in the environment. So examples of biotic factors include predators, prey, so any living organisms, trees, flowers, plant life in general, they are considered living. But it's not just the physical living organism themselves, it's also their actions. So if animals are competing for space, if they're competing for food or mating, this is all considered biotic factors as well. So competition for resources, etc. For the abiotic factors, these are the non-living components in the environment. So examples are things that you will be measuring in this week's lab, such as sunlight, temperature, soil moisture, light intensity, soil pH, etc. All of these things can influence biotic factors. And also, weather conditions like rainfall, hurricane, floods, these are all examples of abiotic, including natural disasters as well. So the first thing to examine is, let's go back and take a look at the biotic factors. So how do biotic factors affect another biotic factor? So how does something living affect something that is also living? So let's go back to the example of competition. Okay? So seals are animals that mate during January to March so that they can give birth later on in the summer month when conditions are more favorable. So there's a very short time period in when they can mate. So seal mating, that's in action, that's a biotic factor, is going to lead to an increase in competition for space, which is also a biotic factor. So seal mating during a small amount of time is going to eventually lead into competition for space, because seals, they have to go on land in order to mate. So as you're studying, make sure that you can come up with your own examples. We can now take a look at how an abiotic factor can affect another abiotic factor. So how can something non-living can affect something that is also non-living? An easy example would be if there is high sun exposures in an area, that's going to lead to a reduction in soil moisture in that air as well, because high sun exposure, high light intensity is going to cause water to evaporate. And another example can be whenever there's high wind, this can lead to a fire. Wind is an abiotic factor. Fire is also an abiotic factor. So high wind, okay, when there's high wind, the wind can carry embers. They can also carry a high content of oxygen. All of these things can facilitate fires. And please make sure you can come up with other examples, your own examples, as you're studying as well. All right, so now let's take a look at how these two factors can influence each other. So first, how can something that is living can affect something that's non-living? So how can a biotic factor affect an abiotic factor? The burning of fossil fuels by humans, so this action that is going to eventually emit a lot of carbon dioxide into the atmosphere. Okay, so burning fossil fuel releases carbon dioxide into the atmosphere. High levels of carbon dioxide, that is an abiotic factor. The burning of fossil fuel is the biotic factor. And then this, we can also take this another step forward, and the burning of fossil fuel will eventually lead to ocean acidity, so having a low pH in the ocean. The reason why is because the ocean is a huge reservoir for carbon dioxide, so the ocean can absorb a lot of carbon dioxide. If there's a lot of carbon dioxide in the atmosphere, the ocean absorbs it, and that's going to decrease the pH in the ocean. So this is an example of how the use of fossil fuel, burning of fossil fuel by humans, which is a biotic factor, can affect abiotic factors downstream. High levels of CO2, that's an abiotic factor. Ocean acidity, that's also an abiotic factor. Now we can take a look at how something that is an abiotic factor can affect a biotic factor. So now, how can something non-living can affect something that is considered living? In this example, we're looking at environmental temperature. So lizards and a lot of reptiles, okay, they are ectothermic, meaning that they cannot regulate their internal temperature, their internal physiology. Humans and mammals, we have homeostasis. If it gets too hot, we sweat. If it gets too cold, we shiver. The shivering of our muscles is going to act as some movement in our body so that we can warm up. Lizards, for an example, they cannot do this. So whenever it is too cold, you'll notice that the lizards are going to go out and sunbathe. Okay? You'll see this with turtles as well in the pond. Vice versa, whenever it's too hot, the lizards are going to move into the shade to stay away from the hot temperature so that they can cool down. So the abiotic factor in this example is the environmental temperature, and the biotic factor is the behavior of the lizards. Another example is looking at animal distribution. So environmental temperature can also affect distribution of certain animals. Tiger sharks, you won't find them in polar oceans. You'll only find them in tropical ocean and warmer ocean because their body temperature their physiological mechanism shuts down whenever it's too cold. So in this example, environmental temperature is the abiotic factor and the distribution of the animal is the biotic factors. Again, make sure you're coming up with your own examples as well while you are studying. So some other examples that we can look at is going back to a very large and important abiotic factor, which is sunlight. In the northern hemisphere, where we are living, the south side of the slope is more exposed. It gets more direct contact with the sun. Okay? So this is what we see in the northern hemisphere. Because of that, the north side and the south side of the slope, they're going to have different variation in life there. So since we are in the northern hemisphere, the south side of the slope gets that intense sunlight hitting it throughout the day. The north side also receives sunlight as well, but just not at that same light intensity level. Because of that, you would expect to see that the moisture level is going to be much more drier on the south side of the slope compared to the north side, because again, the south side is getting that direct contact exposure. This eventually is going to probably influence maybe the abundance and diversity of organisms living there. So abundance is overall looking at the number of organisms. You can look at the overall number of plants. You can look at the overall number of animals, the overall number of certain of flowers, etc. Diversity, on the other hand, is looking at the species richness, okay, the species index. So for an example, abundance, you can take a look at the overall number of flowers. Okay. Diversity, you're looking at the different species of flowers that are found there. Okay. Sometimes abundance, you can have a lot of organisms living there, but diversity is low if you have very different species. So in this example, start to think, which side of the slope do you think is going to allow for more diversity? So here is an actual picture, okay? This is the south side of the slope, this is the north side of the slope. The south side of the slope is the area that's getting more contact with sunlight, so the moisture in the soil is going to be lower. Okay? The north side, the reason why you see it's more green is because the soil moisture is higher. Okay? So with that said, okay, you should be able to determine which side of the slope may allow for more diversity. Okay, so now since we are going to do some data collection, let's review the scientific process. In this example, I have simplified the scientific process down to just 4 steps, but essentially, please understand that the scientific process is not linear. It's actually a circular loop. It doesn't always go in this order. But just to simplify things, the scientific process allows us to observe everyday stuff and come to a conclusion. So, for an example, if you drink coffee and you realize one morning or you observe one morning that your coffee machine is not broken, okay, the first step is the observation. The second thing is you may start to question, okay, why is it not working? Is it not working because I forgot to plug it in? Is it not working because the filter is broken? So these are your questions. Your question eventually leads you to form a hypothesis. A hypothesis and predictions, they are two different things. So for example, the hypothesis is, well, this is just an example. The coffee machine is not working because I forgot to plug it in. That is the hypothesis. Okay? Or the coffee machine is not working because it's not plugged in. That is the hypothesis. The prediction would be, if we plug in the coffee machine, it's going to work. So try to understand the difference between the hypothesis and the actual prediction. Okay. All right, and then after you formulate your hypothesis, you can design an experiment to test your hypothesis. And then after you collect some data in science, you can analyze your data using statistics. So here's another example. Okay. So this is an example taken from Connell, which he was a very famous Scottish researcher back in the 60s, 70s. But essentially, he noticed that when he was walking along the intertidal zone in the ocean, he saw 2 different barnacle species, and the way that they were distributed on the rocks was very distinct. The thalamus, which was the smaller barnacle species, were found at the very top of the rock, so they were more exposed to air. The semibalanus, they were found in the lower portion of the rock, so they were less exposed to the air. They were more in contact with water. So that's just his first observation. He also noticed that the thalamus, they were also the smaller barnacle species, and the semibalanus, they were the smaller barnacle species. So after his observation, this leads to his question. Then he started to wonder, okay, what is the reasoning for this distinct distribution? What's behind it? Some hypothesis could be that there may be some specialized adaptation that can allow for the thalamus to survive in the top part of the rocks and the semibalanus to survive in the lower portion of the rocks. So it could be that each animal had a specialized adaptation. Okay? So another example can be, maybe there are certain tide patterns that would affect their distribution throughout each time of the year. Again, we do not know which hypothesis to accept until we actually design an experiment, collect some data, and then actually test our data using statistics to prevent bias. So Connell did this experiment, and what he did was he took a portion of the thalamus, the smaller barnacle species, and he placed it on a different rock with no other barnacles. And he found that when the thalamus was on an empty rock by themselves, they moved down in the intertidal zone, so they were less exposed to air. So after his conclusion and after other things that he did, he came to the conclusion that the reason why we saw this distinct distribution of the barnacle species is really due to competition. The semibalanus, since they were bigger than the thalamus, they get to actually occupy their specific niche. So they can go and live and survive in the lower part of the intertidal zone, which is actually more ideal for all the barnacle species. And the thalamus, they were forced to live in the upper area due to competition. So competition was one of the main driving factors that led to these distinct distribution. And again, we wouldn't get to that conclusion unless we design our experiment and test it using stats. So in this week's lab, you're going to go outdoors and see how the sunlight, which is a very strong and huge abiotic factor, can affect other abiotic factor. So essentially you're going to get a bucket, okay? Make sure you record down your bucket number, and that's going to be your plot number as well. So make sure you record that here in your data sheet. And you're going to measure soil temperature, air temperature, soil pH, soil moisture, and light intensity. When you go outside, you'll see a plot that is netted so light can come in. This is going to be the sun area. And you're going to see a plot that is darkened. This is going to be the shade. So this represents the sun, this represents the shade. Okay? Inside your bucket, there's going to be a ruler. All you have to do is use the ruler to plot the shade so that it stands up, so you don't have to hold it all the time. One of the things that you're going to measure is light intensity. So we're going to share the photometer reading, and it'll tell you the light intensity under the sunny area and under the shaded area. And the light intensity is in micromolar, the units. But essentially a photometer measure light photons. And as of right now, okay, you can come up with some hypotheses. This is practice 3 in your lab manual. So a good way to write your hypothesis, this is just an example, but let's say you hypothesize that light intensity is higher in the sun compared to the shade. Make sure you include a good rationale. So there should be a because statement. Why would that be? The second thing you're going to measure is air temperature. So for the air temperature, you're going to use a thermometer. Make sure you plug the probe into the correct outlet. This is the thermometer right here. And when you are recording your values, make sure you're using the SI. So it has to be in degrees Celsius. So it'll tell you right here. Okay? And essentially for air temperature, what you're going to do is you're not going to hold this wand in the air. You're just going to touch the area underneath the plot, okay? And that is the air temperature right here. So you're not holding this wand in the air. To measure soil temperature, you do the same thing, except for now you're taking the wand and you're touching the soil. That's all you have to do. So remember, before when you were doing the air, just measure the air temperature between the plot and the grass. For soil temperature, actually touch the soil. You don't have to dig this all the way down, but gently touch the soil. Just the surface area would be fine. Okay. And remember, you're doing this in both the sun versus the shaded plots. For moisture levels, now we're moving into the probe. The soil pH and moisture probe looks like this, and you'll notice that there is 2 black lines here and 2 black lines on the top. This though, you actually have to go deep into the soil. So there will be shovels that you can dig a hole. Okay. So you're measuring moisture level in the sun versus the shade. And when you put this into the soil, make sure that you go up to the second line right here to get accurate reading. For the soil moisture, it's the reading on the very bottom of this diagram right here. So the reading here on the surface, make sure you're looking at the number on the bottom. Okay, it goes from 0 to 100. So it starts with 0 on the right side and it goes to 100, which is on the left side. Okay, and for this, when you are reading the soil moisture, you have to place your thumb on this button right here so that you're reading the moisture level. This arrow, this line, is going to fluctuate. Okay, between 0 and 100. That tells you the moisture level in percentage. So the unit is going to be in percentage. Then when you want to measure soil pH, all you have to do is let go of your thumb on this button. So when you're measuring pH, you don't have to hold on to anything. It's not until you measure soil moisture that you actually have to hold your thumb on the button. For soil pH, just leave it in there and it'll tell you the pH. The pH is going to be the number on top. So it ranges from 3.5 all the way to 8. Okay, so usually pH goes from a range of 0 to 14. Here it's just 3.5 to 8. Okay, so the pH is the number on top, moisture is the number on the bottom. When you're done collecting your data, we're going to go back into the class and we are going to pull our data on the board. So we will have an Excel. This is just a hypothetical. This is just data from the previous semester. Go ahead and put in your data. Okay? Make sure your units are in SI. You should be using the correct units. And your number, the way you round it, make sure you're rounding it according to the lab manual. Okay? So we'll plot our data on the board and then we'll discuss the mean, standard deviation, and standard error of mean next. All right, that's it.